Explore how CRISPR gene editing works, from bacterial origins to molecular mechanics, therapeutic and agricultural uses, and the technical, ethical, and regulatory challenges that will determine its future. You'll learn concrete facts, examples, and how CRISPR edits DNA. Chapter 1: Origins, CRISPR in Bacteria, and the Discovery that Changed Biology. CRISPR originated as a bacterial adaptive immune system, clustered regularly interspaced short palindromic repeats with spacer sequences captured from viruses. First described in 1987 and recognized as adaptive immunity in 2007, CRISPR arrays plus Cas nucleases provide sequence-specific cleavage. The human genome contains about 3.2 billion base pairs, so precise targeting matters. In 2012, Jennifer Doudna and Emmanuelle Charpentier adapted a Cas9 guide RNA pair to cut DNA at programmable sites, transforming molecular biology. CRISPR loci combine short repeats, 20 to 40 BP, and spacers, 30 BP, that record prior infections. This background explains why CRISPR can be guided by a short RNA to find complementary DNA, enabling straightforward reprogramming. Chapter 2: Mechanism: How guide RNAs, PAMs, and cellular repair produce edits. CRISPR-Cas9 uses a guide RNA, typically 20 nucleotides, to base pair with a complementary DNA sequence adjacent to a protospacer adjacent motif, PAM, commonly NGG for SPAC-Cas9. NGG appears roughly once every 16 base pairs in random DNA. Cas9 cleaves 3 bases upstream of the PAM. The double-strand break is repaired by cellular pathways. Error-prone non-homologous end joining, NHEJ, often creates insertions-deletions that disrupt genes, while homology-directed repair, HDR, can introduce precise changes when a DNA template is supplied, though HDR efficiencies are often under 10% in many cell types. Variants, base editors and prime editors, enable targeted base substitutions or small insertions without double-strand breaks. Chapter 3: Applications: Medicine, Agriculture, Diagnostics, and Industry. CRISPR has rapidly moved from lab tool to practical use. Clinically, ex vivo editing of hematopoietic stem cells to upregulate fetal hemoglobin has shown dramatic benefits for sickle cell disease and beta thalassemia in ongoing trials. Patients received a single autologous infusion after conditioning, and many experienced durable symptom reduction. In agriculture, CRISPR produced non-browning mushrooms, 2016, and herbicide-tolerant soybeans, accelerating trait development without foreign DNA. Diagnostics harness Cas12, Cas13 collateral cleavage for rapid nucleic acid detection. Sherlock and Detector can detect viral RNA at attomolar to femtomolar sensitivity in laboratory settings. Industrial biotechnology also uses CRISPR to optimize microbes for biofuels and pharmaceuticals, reducing development times from years to months. Chapter 4: Challenges and Implications: Delivery, Safety, Regulation, and Equity. Advanced challenges determine how CRISPR will scale. Delivering editors in vivo requires vehicles, adeno-associated virus, AAV vectors, lipid nanoparticles, LNPs, or electroporated cells. AAV's 4.7 kb payload limits use of large Cas enzymes, motivating smaller nucleases like say Cas9, 3.2 kb. Off-target edits, immunogenicity to bacterial Cas proteins, and mosaicism in embryos remain technical concerns. Robust assays, GuideSeq, CircleSeq, help quantify off-target sites. Ethically, somatic therapies proceed while human germline modification is widely constrained after the 2018 He Jiankui case. Regulatory pathways, long-term follow-up, and equitable access are critical. Safe, affordable delivery and surveillance will shape societal benefit over the next decade. CRISPR transformed a bacterial immune system into a precise programmable genetic toolbox. Understanding guide design, repair pathways, delivery methods, and ethical limits is essential. Practical applications already span medicine, agriculture, and diagnostics. Future impact depends on improving accuracy, delivery, regulation, and ensuring equitable access.